Okay, welcome back. So in this lecture we are going to start a new module which is genome construction using graph algorithms. So from this module onwards we will start to see some graph algorithms as well. For this lecture I want to give you introduction to the genome assembly problem, that is a genome reconstruction problem, and afterwards we will get into more details of this problem. So far just to remind you that we have covered 4 modules, and from this lecture onwards we are getting into the 5th module, that is genome reconstruction using graph algorithms. So let us get started. So just to remind you, in the whole genome sequencing process we have a target genome sequence which is unknown to us. Now in a typical DNA sample extracted from either blood or saliva, we will have multiple copies of the DNA, right. So there will be many copies of the genome inside the sample, and suppose if the amount of DNA is small, then there are biochemical processes in which we can amplify the DNA and create more copies. So we can assume that in our sample we have several copies of the genome. We will pass this sample through the sequencing instrument, and as we have seen in the previous lectures, the sequencing instrument will give you fragments of the genome as output. So from these copies we will break these genomes into fragments, and then the sequencing instrument will read each of these fragments and give you the output in a digital format. So what we have as that we can see ultimately is these read fragments that we have to now analyze. So we do not have access to the target genome and we do not know the genome sequence. So all we have is the read fragments as input, and these fragments are sampled from random locations of the genome, and with each fragment we do not know the location from where it is sampled from. So for each fragment we only know the sequence corresponding to it. In the genome assembly problem, what we want is, using these reads we want to reconstruct the original genome back. So using the data that we have at the bottom, we want to reconstruct the original sequence back from where these fragments originated, and this is the genome assembly problem. Now, so why is this problem interesting and why is it important? Let us look at that. So first of all, a recap again, that a human genome sequence comprises 46 sequences. We have 22 pairs of chromosomes plus 2 sex chromosomes. So for males that would be X and Y, and for females that would be X and X chromosomes. So if we look at the combined length of all these 46 sequences, the combined length is about 6 billion bases. And also another point that will be useful later is that these pairs of chromosomes are almost near identical to each other. So we inherit one copy of these chromosomes, let us say chromosome 10. So we inherit one copy of this chromosome 10 from father, and the second copy of the chromosome would be inherited from mother, and these are near identical to each other. So there will be like about 99.9% identical to each other on average. So in the genome assembly problem, basically the goal would be, if we are assembling a human genome, then we would like to know the sequence corresponding to the entire genome, that is the entire 6 billion bases sequence that tells me the genome sequence of all 46 chromosomes. So given this property, once we have sequenced the DNA and once we have obtained the read fragments as output from the sequencing instrument, the ideal output of the genome assembly problem is basically 46 sequences, one per chromosome. So, that is the ideal output that we would want from a genome assembler in practice. However, there are several challenges, as we will see in the next few slides, which prevent us. So, this process of converting these fragments into a single sequence per chromosome is not easy. There are several challenges involved, and we will see some of the challenges in the next few slides. So, commonly we also draw an analogy between genome assembly and a jigsaw puzzle. So, remember that in a jigsaw puzzle you have these individual pieces and you have to organize them together, so that you can get the final picture that you are aiming for. In the genome assembly problem, instead of having these individual pieces, you have the reads that you want to put together, and your full picture corresponds to the entire genome sequence, right. So, in some sense there is the analogy between the genome assembly problem and the jigsaw puzzle. However, so because each piece is a read sequence, so now you can imagine that to understand whether the reads come together in the genome, maybe we can consider the suffix prefix overlaps between those reads to understand maybe that, okay, if the two reads have a long suffix prefix overlap, then maybe they occur next to each other in the genome sequence, and maybe that can be a useful property that can be used to reconstruct the genome sequence from the individual fragments. So, this analogy is useful because now I can talk about the challenges of genome assembly from a jigsaw puzzle perspective. So, why is the genome assembly problem challenging and as well as interesting? Well, the first reason why the genome assembly problem is usually challenging is because the genome sequences can be very repetitive. So, for example, you can imagine that if your jigsaw puzzle in your solution, you have these repetitive patterns that are occurring again and again. So, here I am showing you a jigsaw puzzle in which the final solution, it looks like there are many repetitive patterns, like you have these many human beings in this jigsaw puzzle, and now if you imagine solving this jigsaw puzzle, it will be harder for you to figure out where exactly each piece belongs because of the repetitive structure. Similarly, in the genomes you have repetitive sequences, and those repetitive sequences make it harder for us to precisely give out the correct order of the reads in the genome sequence. Okay. So, that is the first challenge. The second challenge is that we have too many pieces in the genome assembly problem. So, that is the number of reads that we have to assemble is usually in millions. So, for example, if you are assembling a human genome, then you would be working with millions of reads that you would have to piece together. So, again that makes the problem challenging. The third challenge is that we can have missing pieces. So, we are not guaranteed that all the pieces are there available to us. So, for example, it may happen because the genome sequence is very long, it may happen that we do not have any reads covering a particular region of the genome. So, or we may have insufficient coverage at some regions because the genome sequence is very long and the sequencing process is random. So, the reads are uniformly sequenced at random from the genome, and it can happen that there are some regions where you have insufficient or low coverage in terms of the reads. So, when you have insufficient coverage, that means that you have fewer number of fragments to support that region of the genome. Fourth challenge is that many pieces can be damaged. So, for example, the sequencing process is not perfect. The sequencing instruments can make errors in the sequencing process. So, for example, when we talked about sequencing, maybe the actual sequence was CAGT and the sequencing instrument made a mistake and converted this A to C. So, the sequencing instrument itself can make mistakes in producing the data. So, when you have sequencing errors in the data, that is equivalent to saying that some pieces can be damaged, that is there can be sequencing errors in the reads that we are working with. The final challenge is that when we are solving a jigsaw puzzle, we usually know the final solution that we want to assemble. So, you will have access to what the final picture looks like. However, in genome assembly we do not have a reference to work with. So, we only has this read set as input, that is we are only working with these read sequences, and we do not know what the final solution is going to be, because that is what we want to know ultimately, right. So, only reads are given as input. When we do not have access to the final solution, then naturally this jigsaw puzzle becomes harder. So, with all these challenges in mind, this is why genome assembly problem has been a very challenging problem to solve for many decades, and over the years progress is being made to progressively address these challenges. Why is this problem important? Well, it is useful for answering many fundamental questions in biology. For example, when we want to build high quality reference genomes, or we want to know the genomes of novel species for biodiversity purposes, we may be interested in reconstructing metagenomes, that is a collection of microbial genomes that are available in some sample. For example, if I sequence a water sample or if I sequence some soil sample, I may want to know the complete genome sequences of the microbes that are present in that sample. Similarly, I may be interested in studying genome evolution, like how the genome sequences are evolving over the time period, and also because genome assembly gives you the entire genome sequence as output, I can also study chromosomal rearrangements, duplications and inversions in the genomes. So, these large structural changes can be studied when you have the entire genome sequence available to you. And this is because of these applications, the genome assembly problem has been a fundamental problem in bioinformatics for many years. Let me show you also some history of this problem. So, this I would say that genome assembly problem is about 50 years old. In fact, the first attempt to assemble a genome was in 1977, when a small genome of length 5000 bases was assembled. So, this was a virus genome that was assembled in 1977 using Sanger sequencing. Now, for coming to 2 decades later or 2.5 decades later, the human genome project was completed, and now you can imagine why it took so long, because the human genome sequence is significantly longer. So, compared to genome length of only 5000 bases, human genome is 3 billion bases long, right. So, if you consider only a single copy of each chromosome or a haploid sample, then the entire human genome sequence is 3 billion bases long, and therefore, the amount of effort in terms of the data generated and data analysis is significantly higher for human genome compared to a virus genome. So, this human genome project was completed in 2003 with an estimated cost of $2.7 billion, and the first draft human genome sequence was released at the end of this project. So, this was a major milestone. Following that, the sequencing cost has dramatically dropped. In fact, in 2014, the cost to generate a high quality human genome sequence reached only $1000. So, this is a significant jump from $2.7 billion to only $1000. So, again this can be a major milestone. And one of the recent milestones in the genome assembly problem, it has been the first complete and gapless human genome assembly. That is assembly where there are no gaps and you have exactly one sequence per chromosome. That is the ideal output that I was mentioning earlier. So, this was achieved for the first time in 2021. So, that is the first complete and gapless human genome assembly was published. So, these are the recent milestones as far as genome assembly is concerned. Now, that covers the introduction of the genome assembly problem, that is what genome assembly problem is, that is we are trying to use these read fragments and trying to get back the original genome sequence. So, in other words, we are trying to reconstruct the original genome sequence from these short read fragments. And with this introduction given, let us get into the technical details now. So, first question that we can ask is, how much sequencing data is enough for genome assembly? Because when I am doing any sequencing experiment, what I have in control is how much data I want to generate. So, for example, the number of reads that I want to generate from a genome is under my control. I may say that I want to generate 1 GB data, I can say I want to generate 10 GB data, or I want to say 100 GB data, so on. So, a natural question arises is how much sequencing data is enough for doing genome assembly, right. So, that is one question that we will address. The second question is that we will study in the next lecture onwards is, suppose now you have generated enough data, let us say you have generated enough data that you think should be sufficient for genome assembly. Now, once you have the data, how do you generate the entire genome sequence? And there we will look at some interesting graph theoretic algorithms that will be useful in the genome assembly problem. So, that we will leave for the next lecture, and for this lecture we will focus on this question of how much sequencing data do we want to generate to assemble the genome. So, as I said, the amount of data that we want to generate can be controlled by the user, because during the sequencing experiment you can specify in the instrument how much data you require. So, let us see one attempt to answer this question, and before we get into the details, I want to convey one definition, that is coverage. And coverage is an important term that is used commonly in the genome assembly literature. How we define the coverage is as following. So, coverage is defined as the number of reads covering a position of the genome on average. That is, in other words, you can think of it as the average redundancy in the sequencing data, right. So, normally we want to ensure that there is redundancy, because we are going to look at suffix prefix overlaps among the reads. So, that naturally implies that we need some level of redundancy in the data, so that we can assemble the genome accurately. So, for example, I can say that if I am sequencing at 30x coverage, 30x coverage implies that each locus of the underlying genome was read out 30 times on average. That is, each locus of the genome was read 30 times on average in the sequencing data set, right. So, that would mean that on an average each base of the genome is read 30 times in my input data. So, in this particular example shown on the left, it is easy to calculate what the coverage should be. So, for example, the way I can calculate the coverage is as following. So, you can basically calculate the total number of bases in the reads. So, if you look at all the reads here, and if you add up the number of bases in all the reads, you will find that the complete read set has 177 bases, and the genome sequence has 35 bases below. So, the coverage for this particular example would be 177 divided by 35, that is about 5x, suggesting that each base of this genome on an average is sequenced 5 times in the read data set, right. So, accordingly, like higher coverage implies that you have more number of reads basically. So, a trivial exercise can be that suppose you have read length 100, that is all reads have length 100, then how many reads would you need to sequence for the human genome to achieve 30x coverage. So, if your read length is fixed to be 100, then how many reads do you need to sequence from a human genome to achieve a coverage of 30x. So, this is simple to address because again you can apply the same formula, because 100 is a read length and suppose n is the number of reads that I want to sequence and my genome length is 3 billion and I want to achieve 30x coverage. So, it immediately gives me the value, that is 90 times 10 raised to power 9 divided by 100. So, such that n is equal to 9 into 10 raised to power 8. So, that is roughly you will need 900 million reads of length 100 to achieve 30x coverage on human genome. So, this is to just iterate the same fact again, that when we are working with human genome data, especially for genome assembly problem, we are usually working with very large amount of data, and now you can imagine that reconstructing the genome from millions of reads is not an easy problem, and that is what makes this genome assembly problem interesting. So, I hope the definition of coverage is now clear, that it is indicating the redundancy in the data, right. So, and if we sequence the genome at 30x, then on an average we are saying that a position of the genome is read out 30 times on average. Okay, so let us proceed. Now we can answer the main question that we want to ask. So, how many reads do we need to cover the entire genome? So, you have the genome sequence, you have the reads sampled from random positions of the genome, and how many reads do we need to cover the entire genome. So, let us say we have length of each read is equal to L, that is all reads are of fixed length L, let us assume that, and also let us say that when we are assembling the genome, we want to find some suffix prefix overlaps among the reads. So, let us say that the suffix prefix overlap that I am finding between the reads should be of at least length theta L, that is theta is conveying the fraction of read length that I want to see in the overlap. So, for example, if I want to see at least 15% of the read length in the overlap, then I can set theta equal to 0.15, suggesting that I at least need 15% of the read overlapping with a second read, right. This is to ensure that we do not, because we do not want to allow very small overlaps, right, because those small overlaps can happen by chance as well. So, normally you want to enforce some threshold on the overlap length as well. So, that can be 15% of the read length or 30% of the read length, that is up to you. So, let us denote that theta indicates the fraction of the read length that you want in the overlap, and L is the length of the total read. Okay. So, now let me define another term which is called as island. So, island is a contiguous group of reads that are connected by overlaps of length greater than or equal to theta L length. So, that is theta L is the threshold that we are considering, and so these are the group of reads that are consecutively overlapping with each other. This is visualized in the figure below, where you can see that this is one island where you have 3 reads overlapping with sufficient threshold. In the next you see that maybe here you have all the reads overlapping with each other consecutively, but this overlap is not sufficient for us. So, therefore, this becomes a second island, and this becomes a third island. So, this illustration shows you an example where you have 3 islands, that is 3 groups of reads that are connected by overlaps of length above the threshold. Okay. So, now we can ask what is the expected number of islands that we get, assuming reads are sampled uniformly randomly from the genome sequence. Ideally, what we want is that we want to see the reads overlapping with each other and such that they cover the entire genome sequence. So, that is why we want to study the expected number of islands, so that we can understand how this expression changes with the number of reads, that is, so that we can control and we can ask for appropriate amount of data, so that we can ensure that the reads cover the entire genome sequence. Okay. So, we will in this analysis let us derive this expression, and for this analysis I am going to use some basic notations, that is I will assume that n is the number of reads, L is the length of each read, G is the length of the genome sequence, and C denotes the sequencing coverage, and that will be basically as we defined before, it will be n, that is the number of reads, multiplied by length of each read divided by the genome length. So, that will be n L by G. So, with these notations let us derive the expected number of islands given these parameters. So, let me derive this by hand. So, let us say, so you have this genome sequence, and out of this genome sequence n, there are n such positions in this genome sequence where you have a read starting, because you have n number of reads. Now, let us indicate, let me define a variable lambda which indicates the probability that a read starts at a given position. Right, and this will be simply, so lambda in this case will be n by G, because you have n positions where the reads are starting, and because the read starting positions are random, the probability that a read starts at a particular position in the genome is n by G, where n is the number of reads and G is the genome sequence length. Now, let us, based on this, let us look at probability that k reads start in an interval of length x. Right. So, basically this is equivalent to saying that you have x number of trials and you want at least k successes, right, and the probability of success is small, that is the probability of success is n by G. So, you want at least k successes in x number of trials. Now, the expected number of successes is x lambda, right. So, you have the interval length is x and the probability of success is at a particular position is lambda. So, when you have the entire interval of length x, then the expected number of successes in the entire interval is x lambda. Now, assuming that lambda is small and assuming that this success probability is small, we can make a Poisson approximation to the binomial distribution, and we can say that probability k reads in length x equals, applying the Poisson distribution, is equal to e to the power minus lambda x to the power k divided by k factorial, that is applying the Poisson distribution formula, assuming that the mean number of successes is lambda x, right. So, based on this, now we can proceed forward. So, now we can derive the expected number of islands in the following manner. So, remember that the expected number of islands is equivalent to saying that you have n number of reads and probability that read is at rightmost end of an island. So, basically you have an island whenever a read is the rightmost end, that is if there is a no read following it further, then you know that you have a island, new island formed by it. So, that is you have a read in which you have no further read that is having sufficient overlap with this read. So, all the reads are either not having sufficient overlap or they are far apart. So, that will convey you the expected number of islands, because now we can look at each individual read and check what is the probability that that read is the rightmost end of an island. So, given this, basically this is easy to calculate now. So, this is n times probability of 0 reads starting in the interval of 1 minus theta L, that is we do not want any read to start. So, if this is let us say theta L, then I do not want any read to start in this 1 minus theta L interval, because only then this read will be the rightmost end of an island, right. So, I do not want any read to start in this particular interval. So, that we can calculate using the formula that we have in the previous slide. So, applying that, putting k equal to 0 in this formula, what we get is e to the power minus lambda so putting the value 1 minus theta L, that is the interval length, and then we want 0 successes. So, this becomes the expression from the previous slide. So, now simplifying this further, what we will get is minus lambda 1 minus theta L, right, and I can further rewrite this as, so I can place the value of lambda, and putting the value of lambda we get minus 1 minus theta L n by g, right. So, now what we can do, so this is the expected number of islands that we will get if we have n number of reads and L is the length of each read and G is the genome length. So, how does this expression look? Well, let me also do one more modification. Let me express this formula in terms of the coverage C, because it is easy to interpret the coverage parameter rather than the number of reads parameter, because the coverage is easy to interpret. So, let me rewrite this as, so remembering that the coverage is C equal to Ln by G, right. So, I can express this formula in terms of coverage as well. So, if I do this modification, I can rewrite this as n e to the power minus 1 minus theta c, and again I can replace n with, so n equal to c g by L. So, this becomes g by L c e to the power 1 minus theta c. And now I can ask, okay, as I keep on increasing the coverage of the data set, so because my genome length is fixed, my read length is also fixed, and now I can ask, okay, if I keep on increasing the coverage, let us say I sequence at 1x, 2x, 10x, 15x, what is the expected number of islands I get, and until what point do I ensure that I can cover the entire genome sequence, right. So, if you plug in some values, so if you I can plug in some as an example, I can assume, let us say that my read length is 1000 and let us say that my genome length is 1 million bases, and theta is I can assume different thresholds. So, for example, as we discussed, you can have different thresholds of the overlaps that you want. So, if you want 15% overlap among the reads to connect them, then this kind of curve is what you get. So, this is the curve indicating the expected number of islands on y axis and coverage on x axis. So, is this intuitive? Well, yes, because imagine you have the entire genome. What this plot is telling you is that starting in the beginning, as you keep on sequencing reads. So, suppose if I keep on sequencing reads, initially the number of islands will increase, which makes sense because initially all the reads may not have their neighbor overlapping read, but at some point when I have sufficient coverage, then the number of islands will start to reduce, and this is what this expression is showing. And also, if you want higher threshold, like if you want higher overlaps among the read lengths, then you would automatically need higher coverage as well. So, if I desire higher overlaps versus smaller overlaps, then at higher overlaps I will need higher coverage. So, this plot roughly indicates how the trend follows, like at how the coverage dictates the expected number of islands, and ideally we want to sequence the genome at a coverage where the expected number of islands is close to 1, right. I should mention that this plot cannot be trusted at very high coverage values, because if you plug in very high C values in this expression will approach 0. So, it will become less than 1, which does not make sense, because the expected number of islands will always be an integer value, whereas if you put in very large coverage values, this expression will approach 0. This, which is because we are making some approximations in this expression. So, for example, binomial to Poisson approximation, and those approximations will not work if you have very high coverage data. So, to summarize, this expression shows you how varying coverage gives you the expected number of islands, and then we can accordingly ask for enough amount of data to be sequenced from the genome to do genome assembly. So, in this lecture we looked at the introduction of genome assembly problem, and we also looked at what are the major challenges involved, what are the major milestones that have been occurred so far. Also, we looked at one technical detail of how much data should we generate to assemble a genome, that is how much coverage do I need over the entire genome sequence, so that I can make sure that the reads cover the entire genome. So, in the next lecture we will now assume that we have generated sufficient amount of data, and we will look at some of the common algorithms that are used in practice to assemble genomes. Thank you.